Bioinformatics.
Okay, in this lecture, I'm going to be talking about some of the basics of gene expression and how to compare gene expression using some of the genomic and bioinformatic methods we've been using throughout the semester between species, both quantitatively and qualitatively. So the goals for this, really, kind of the take-home for this lecture, are to understand the difference between a qualitative and a quantitative experiment in the context of gene expression comparisons, how to understand what we actually mean when something is differentially expressed, and also how to think a little bit about proper experimental design for quantitative expression studies. The reason I focus more on this than some of our other methods we've talked about in the past is because in my experience, many of you that take this class, although there's a good chunk that won't use these types of methods in your projects, those of you that do are more likely to use differential expression analyses than just about any other comparison. So I like to provide more context there. So first thing I like to clear up is we often abbreviate this as RNA-seq or RNA sequencing, but in almost all studies that you look at, now there are plenty of exceptions. There are microRNAs and all sorts of things, but many of the studies, particularly eukaryotic, of course, where by saying RNA-seq, we're actually referring to mRNA sequencing. We're almost always comparing RNA at the mRNA level. And so not primary transcript, but something that's actually already been through some processing. Now, historically, there are different ways to look at gene expression. There are kind of the more traditional real-time PCR approaches where you're looking at the number of transcripts for a single gene, both in a wild type versus a mutant or two different treatment categories. And this is very low throughput because you're looking at one gene at a time. If you were working in a model organism, a little bit later on, microarrays were developed, where essentially there was a well in a big giant plate for just about every major gene in some model organism. And you would take individual samples of that species or closely related species, maybe between one that was subjected to heat and one that wasn't, and you would basically tag all of the RNA from each sample with a different color, essentially. And so what would happen is at the bottom of each one of these wells for any given gene, there were a certain number of places for those RNAs to go and bind. And once it got filled up, no more could bind. And so if it was all red, then that would signify that just one of the species had transcripts that were binding for the most part. If it was yellow, in between green and red, you would see that there was almost a 50-50 binding of transcripts from each of those two different samples. This basically reflects kind of the ratio of transcript abundance between the two samples. Now, it's not perfect. You don't get really numerical values per se. You get color intensity that's translated to to numerical values, and you don't get any information about different isoforms and things, but this was used for a long time. Finally, RNA-seq, which is what we use most days now, is really nice in that you don't need any prior information to do a basic differential expression study. You don't need a reference genome. You don't need a microarray already built. You can build a reference transcriptome from the data you're generating to do the differential expression comparison. And so what's great about this is it's non-model friendly. It's numeric. It's based on, as we'll see, read count distributions. And depending on how you sequence it, you can actually start to distinguish variances between different isoforms as well. So these methods are incredibly popular. I did a quick RNA-seq profiling search on just Google Scholar, and this was actually a couple years ago I did this, but there were almost 18,000 results of papers within just the last several years that had RNA-seq profiling as one of their search terms. So this is probably one of the most published forms of genomic analysis. If you're interested in playing around with RNA-seq data, well, there's always the SRA where the raw data are housed. But you can also go, and we talked about this before, the Gene Expression Omnibus is a great place to go and kind of play around with stuff. They have both microarray and RNA-seq data. And then if you are looking for data from European experiments, you can go to Array Express as well. Okay, so let's talk a little bit about the two fundamental different ways of thinking about differences in gene expression. So let's say we were looking at two different treatment groups, some plants that we watered really well and some plants that we didn't. What are some ways that we could compare these things, how they're responding to this drought or water treatment, however way you want to think about it, at a molecular level? Well, we could look at what genes are being turned on or turned off between these two. In other words, if you were to think of all the genes that are on in group A and all the genes that are on in group B, we could create a Venn diagram of sorts to see which genes are on in both of them and which genes are on only in one of the treatments and start to make inferences based on this. This is what I would call a qualitative comparison. We're not looking at how on the genes are. We're just looking at a binary. Are they on or are they off? Conversely, we could look at them quantitatively as well. You could have genes that are on in both treatments, but they might be on more in one treatment. In other words, expressed at a higher level or differentially expressed. And so here's an example where we've got this gene X and this gene Y in two different colors. And here we can see gene Y and gene X are on in both of our groups. Gene Y is not differentially expressed. It is expressed at the same amount in both of our groups. However, gene X is expressed at a much lower level in group A versus group B. This is what we would call a differential expression. So the basic workflow for doing a qualitative comparison is relatively straightforward. You would collect and sequence mRNA, library prep, all that good stuff. You'd do some basic cleanup, maybe clean your FASTQ files, and then do things that you guys have already done before. You would map them to an annotated reference. In this case, that reference could be genomic in nature, or it could be a reference transcriptome that you've assembled from those very RNA that you sequenced. That's a little beyond the scope of this class, but it's something that you can do. You can then create lists, in other words, a Venn diagram of genes shared between these groups and that are unique between these groups. Examples of things that let you visualize this are gene Venn. And then downstream analyses could be anything from you just thinking about it, from you taking lists of gene IDs and thinking about what functions they have and looking for patterns there as well. But really, this is kind of the basic workflow of a qualitative comparison. The advantage of this, typically few, if any, replicates are required to see if something's on or off. That's not something where you need to be very precise about the variability and number of transcripts. It's easy to understand these results. If a gene involved in drought response is turning on only in a drought treatment, well, that makes a lot of sense to us. The disadvantage here is that not a lot of biological processes operate by a strict on and off system. Many of them are mediated by kind of perturbations in the volume of expression rather than just shutting off or turning on completely. And so here's an example of a paper that took a qualitative approach using RNAseq. And in this, they were looking at responses to temperature and drought in some important canola seed crop. And you can look at, they took kind of this Venn diagram approach and looked at across these different groups, which genes were being qualitatively changed. So what are on and, across the treatment and what are only on in certain groups. And, they made some interesting inferences related to functional categories that these different patterns belong to. And you're welcome to kind of go dig through this paper if that's something that interests you. However, we won't be focusing very much on qualitative comparisons. In fact, we're really, from this point forward, we're going to move on. That's pretty straightforward. And we're going to focus most of our attention now on quantitative comparisons of expression level using RNA sequencing. And so the basic workflow for this is we want to make this kind of comparison for every single gene in the genome. And so to do that, same thing as before, we collect and sequence mRNA. We map it to an annotated reference, whether it's a genome or a transcriptome that you assembled from those sequenced RNA. And then we use another software to pull out the number of reads that mapped to every annotated section of that reference. And I'll show you what all these things look like, but we'll take those reads and then we will do some what's called read normalization and then conduct some statistics on it. And that's how we'll get up our initial differential expression result. So I'm going to walk through all of that here in a moment. But before I do that, the general advantages of a quantitative expression workflow is that it's much more powerful than qualitative. Again, most biological mechanisms are not simple on-off. They're levels of on or levels of decrease. One of the big disadvantages is that you need biological replication to really capture the variance in expression level within and between groups as well. And that can get quite expensive. So if we want to talk about replication, because this is an important one when we think about experimental design, most of you are probably familiar with this idea of biologic versus technical replication. In almost all cases, we avoid using technical replication. So here's an example of biological replication. If we wanted to compare leaf versus root expression and see what genes are involved in making something a root versus making something a leaf, we could have biological replication by having 2 biological replicates for each leaf sample. So here we've got a leaf sample from one individual and a leaf sample from another individual, and then 2 bioreps from root tissue as well. Alternatively, we could have technical replicates where multiple leaf samples from the same individual, multiple root samples from the same individual. Without getting into too much detail, in almost all cases, it is the better choice to increase your biological replication as far as RNA-seq is concerned than to increase your technical replication. And this is important because money is very tight with these types of analyses because they can be tens or 20s, thousands of dollars. Speaking of money, estimated costs for something like this. If you want to do a very small study with one factor, so water versus no water, you basically want to have at least 50 million reads sequenced per sample, and you want to have 3 biological replicates for each treatment. So if you're thinking about 3 watered and 3 drought with 50 million reads each, you need about 300 million reads worth of sequencing. That's basically the bare minimum for doing a very small scale differential expression comparison. Okay, so these numbers are always changing, but with that kind of example I gave you where we needed 300 million reads, well, a run of RNAseq for single N will get you about 400 million reads, so that's pretty ballpark what we'd need for that really small experiment. If we did the shortest possible reads, which is the cheapest route, it would cost us just shy of about $2,000 for the sequencing itself and about $100 per sample, so another $600. We'd be looking at around $2,500 or so to do that really basic bare minimum differential expression comparison. Now keep in mind, if you add in more factors or you want to have a better set of biological reps, like 8 bioreps, all of a sudden these things balloon in cost. Now, I mentioned bioreps are really important. You're not always going to see papers following that. Sometimes people really want to do an experiment no matter what, and even if they don't have the funds to do it properly, they'll try and find ways around it. So you will see out there where people do differential expression comparisons without proper biological replication. And what they do is they assume that the expression level variance is similar across all loci. So rather than figuring out what the variance in expression level is for every single gene using your bioreps, they assume that gene X and gene Y and Z all have similar levels of variation, which is a very big assumption and probably biologically incorrect. But that is something that you'll see out there. Okay, so with that background out of the way, let's talk about how these things actually work. How do we use read mapping, like BWA or bowtie, to actually get an estimate of transcript abundance in the cells that we originally extracted RNA from? So the way this works, here we can think about having either a reference transcriptome where we've got just exons represented, or a genome where there's introns and things as well. Whenever you map reads, right, every single one of those little reads finds a home somewhere on your reference. And there'll be different numbers of those that find homes to any given area. The depth, in other words, the number of reads that are mapping to a region, so exon A here has, 5 or so reads that map to it, are analogous to the abundance of transcript prior to sequencing. Because if you think about a cell as basically housing tons and tons of mRNA, When you lyse it open during RNA extraction, you take out all those RNA. And then you dice them up during library prep, put adapters on them, all those sorts of fun things, and sequence them. When you dice them up, you're going to end up with more pieces of the transcripts that were in higher abundance in the cell to begin with, because they're just more there to begin with. And so as you sequence, things are sequenced at random. So the things that are more frequent in your sample and in your library are going to be sequenced more. And so there'll be more of those reads to map to a region. In other words, a gene that is expressed more highly is going to have more reads for it sequenced because it's in higher abundance when you do the sequencing. And so we just work backwards. We go, okay, well, how many reads are in this region? How many reads are in that region? Are there more in this one? That one has a higher expression level. So many of you are familiar already with different read mapping software. BWA, Bowtie. We didn't talk about Top Hat or Salmon, but those are also really nice options as well. There are different issues that can arise during read mapping that are problematic in many analyses, but are extra problematic when you're thinking about estimating transcript abundance, particularly multi-mapping errors. If you have a read that maps to more than one place in the genome or the transcriptome, maybe a conserved sequence that's in multiple genes, well, now you have a problem, because which of those genes was actually being expressed and represented by that read and which wasn't? And so there's different ways to deal with this, everything from throwing out multi-mapped reads to counting them as a fraction. So if it mapped to 3 areas, each area gets a third of a hit, basically. There's no perfect solution, though. So once you've mapped your reads, though, you can use any number of pieces of software to pull out the number of reads per feature or per gene. Software like HitSeq or Express are examples of that. And what you ultimately get is kind of a table that looks like this. This is very simplified, where you have all of your gene names or features, however they're named, and then counts of reads that map there. So for this gene X, there were 250 reads mapping to it, gene Y, 100 reads, and gene Z, 500 reads mapping to it. And you would do this and make a table that represents all of your samples. So going back to our root and leaf comparison, we had 2 bioreps for root and 2 for leaf. So we have 2 individuals for root, 2 individuals for leaf. They all have gene X, whether it's expressed or not, and some estimated read count abundance. That's an analog for expression level. This is the data that we are going to be basically doing statistics on. So we are statistically going to test each gene and say between group A and group B, or the root group and the leaf group, is there a significant difference in these numbers between those groups? In other words, is there a significant difference in the expression level? Differential expression. Things we need to account for when we do this. The plasticity of expression. You could take twins, subject them to the same treatment, and every cell in their body is not going to produce the exact same number of transcripts for any given gene. There is a lot of just variability here. Some of it is due to variation between individuals. Some of it is just unpredictable biological variation. And so this being a 250 versus a 200 is part of that. There's also sequencing bias. Yeah, we're sequencing these fragments at random, but just because you're doing something at random in large numbers doesn't make it 100% accurate at carrying over the ratios. So you will see lowly expressed genes that are slightly overrepresented in their mapped reads than they actually are in the cell. A lot of it, if you have enough bioreps, will get washed out because every individual isn't going to be sequenced biasedly in the same way. So this is where biological replicates become very important. And then we're going to do this kind of statistical test between these. But one of the problems is, those of you that are familiar with the idea of multiple tests, if you've got 35,000 genes and you go through each gene and use some kind of cutoff of a p-value of 0.05 and go, not significant, not significant. Well, think about what a p-value is. It's the percent likelihood that if you reject the null, that you would be wrong. And so that's only a 5% chance. What's 5% of 35,000 genes? That's how many genes you would incorrectly say are differentially expressed when they aren't, if you didn't apply some fancier statistics to avoid what we call false discovery. Thankfully, you do not have to do all these stats by hand. You don't have to go through all 35,000 rows. There are lots and lots of suites of differential expression analyses, and they do things in different ways. They excel in different ways, and we're not going to pick all of them apart. But some of the most popular are DESeq and EdgeR. There's also this VOOM package as well. The kind of unifying theme here is that almost all of them are R packages. And so for that reason, you're going to be using R for the duration of this part of the class. There is a bit of a movement to start getting some Python-related gene expression comparison tools out there. But by and large, once you get that table from your mapped read counts, you're going to import that into R, and that's where the rest of your study is going to happen. So this will be a little different. I'm going to show some snippets from the code that you guys are going to be executing in lab. And you may not have done this yet, which is totally fine. You're going to be doing this soon. But I want to show you the different parts where things are happening, and then show you a little bit about fundamentally what's going on. And so first thing you're going to do, if you haven't used R, is you're going to take that count matrix of all those mapped read numbers, and you're going to read them into R. And so here we've got all of our gene IDs, and then here are our 4 biological samples with 2 bioreps for 2 different treatments, basically. And you can see some genes have no expression. They're just off across all our samples. Some genes seem to have very clear differences, and some do not. So here's where you can kind of see all of your reads, and you're eventually going to tell it what your experimental design is. In this case, the first column was a knockout for a certain gene. The second column was a control. The third column was a knockout for a certain gene or transcription factor. And the fourth was a control. So one of the first things you're going to have to do is something called read normalization. Now, the reason you have to do this is because not all samples on the sequencer are sequenced at the same depth or sequenced by the same amount. And so here, if you think of these as representing the total number of reads sequenced for 2 different samples in the same run, sample 1 and sample 2, let's say this one had 4 million reads and this one had 6 million reads, you couldn't just look at the number of reads from this pool that map to gene A and compare them directly to the number of reads that map to gene A in this sample, because you're pulling from a larger pool. It's not a fair comparison. You can't just go, well, 8 mapped here and 12 mapped here, so sample 2 had a higher expression of gene A. It's just not a valid way to think about things. And so the way that we correct for this is we can normalize the reads by basically, in a really simple sense, there's more complicated ways to do this, but in the simplest sense, you could convert your raw read count to basically the number of reads or number of transcripts they're standing for per million. And so again, if we're thinking 4 million reads were sequenced for sample 1 and 2 for sample 2, or 6 million for sample 2, you could take 8 counts and divide it by 4 million, or 4 if you're doing per million. So there are 2 transcripts mapping to gene A per million of reads in your data set. So you can think about this as a concentration. In sample 2, there were 12 reads, and there were 6 million reads sequenced. And so there were 2 transcripts, or reads, per million in your pool. In other words, there's no difference in the expression level between these 2 samples when you account for the difference in sequencing that happened. And this is a very, very important thing to keep in mind, because you're never going to be comparing totally equal-sized pools of reads. If you're comparing things that vary in size, so say you've got genes that are longer or shorter than one another, well, things that are longer offer a bigger target for reads to map to. And so if you're a longer gene or a longer variant of a gene, you're going to, by nature, have more reads hit you than your shorter comparison. And so that's also something that in cases where that's possible, particularly comparing expression between different types of genes, you can account for that by not only dividing by the number of million reads, but also by the number of kilobases that are the length of that gene area. And so here, let's say we're comparing sample 1 and sample 2. We could see here that we have 12 reads mapping to this red section here, and we've got 24 mapping to this green section here. And so 24 is a lot higher than 12, But if you account for, and this is from the same sample, so the same library size, the same number of reads, but this is just looking at size difference really. If you factor in size difference, you'll see that they're almost the same number of transcripts per kilobase per million, or fragments per kilobase per million, or reads per kilobase per million, however you want to think about these things. Thankfully, again, you will never handle this normalization by hand. I just think it's such an important thing that I wanted you to understand it before you just turn it over to the machines. Doing all of that in DESeq is as simple as taking your data set, here I've named the data set DDS, and doing estimate size factors. So size factors are basically looking at the total number of reads and adding that in for comparison. And so all we're going to do is we're going to estimate the size factors of DDS and then save over DDS with that output. We're then also going to do this thing called estimating dispersions. Dispersions are basically looking at the variance in expression level between your different genes, the whole reason we used biological replicates. So when we're estimating the dispersion, that's basically a way of looking at expression variation or read variation as it relates to inferring expression level. And so there's really two underlying sources of variation here. There is biological noise or variation, which is basically different samples are going to have different numbers of transcripts being synthesized. And that's real variation, and it's important to know. There is also technical variation. In other words, variation related to the process of sequencing, both through sequencing at random and just by chance sequencing certain samples slightly more than others for just those certain genes. So you can get variation in gene expression estimates just due to the random nature of this process. Luckily, without getting into too much depth, if we look at the mean expression level or number of reads mapping to a given region versus the coefficient of variation, it's pretty reliable for us to calculate and look at biological noise. In other words, real variation. And it is really up until this point here, when you get below 100 mapped reads, the primary source of the variation. Technical noise, this blue line here, makes up very little of the total variation until you get below 100 mapped reads. Luckily, at that point, well, 100 mapped reads really isn't very much, and so our statistical power is going to be weak anyways, but technical noise follows a Poisson distribution. And so we do not need actual technical replicates to infer the variance of our samples below a certain number of map reads. It's very easy to basically model it following this Poisson distribution, which you can read more about if you're interested in. In other words, if we think about the total amount of variation, in orange here is related to biological variation, purple to technical noise. Pretty much it's only really about this point here at around 100 map reads that we start to shift over to the majority of it being due to technical noise. And again, that is very easy to predict using a Poisson distribution. And so this is why adding biological replicates is so important, because most of your genes that are expressed highly enough that they're of value to even compare statistically, they are going to be totally determined by their biological variation. So finally, you're going to actually run what's called a negative binomial test if you're doing using DESeq. This is basically a way of incorporating both that Poisson distribution as well as how it levels off to account for biological variation as well. And it's going to compare statistically whether there is a difference in the number of reads that mapped following normalization between your two different groups for every single gene. You can then, and you'll do this in the lab, make a very basic plot called a MA plot. This is a nice way to visualize what's going on. And so what this plot is this is mean expression level for every gene on the x-axis. So everything from basically 0 all the way up to 10 to the 6th power. So really highly expressed genes. Now, on this y-axis is the log-fold change in the mean between your groups. So basically above 0, every single one of these dots across this whole plot actually represents a different gene. And so let's take this gene here, for example. This gene here is expressed at about, on average, 10 to the second mapped reads, okay? And it has a log-fold change of a positive 3 or so. And so what that means is that there is 3 log-fold difference in expression level between the leaf treatment and the root treatment, basically. It's much more highly expressed in the leaves than the roots, depending on how you ordered them when you did your group design. Anything that is significant, in other words, past the statistical test, is in red. So what we can see here is that the higher expressed your genes are, the less difference you need to be able to pull out a significant difference between them. Because you've got more reads, you're better able to account for variation as well, and variation is a lower proportion of the total reads. When you get down to things that are lowly expressed, remember I said things below 100 become a lot trickier. You very quickly need a huge difference in expression level to be able to pull things out. So here you can see all of these seem to have a very large log-fold difference, but we don't have the statistical power to say it's significant. You have to go all the way down here, basically. And so in many cases, these are things where they're basically barely on at all versus being on at a moderate level. And so now, with that just really cursory introduction, you're actually going to go through and do all this in the lab. And you're mostly going to be paying attention to these 3 parts of your results and doing some other stuff with them. Specifically, all your different genes. It's important to know what genes are different because we could actually go and look these genes up on NCBI and see what they do. How they're changing, whether they're positive and increasing in group B versus group A, or if there's a negative log-fold change, meaning that group A is higher than group B. And then the p-value, whether there's a significant difference or not. And so when you're done with the lab, we're going to come back and we're going to have this laundry list of genes that are significantly different between our 2 treatment groups. And we're going to talk about what we can do with that list to try and think about some kind of biological reasons for them being different, just like you would from a real experiment. Okay, thank you.